everyone, and welcome back to this next part of Chapter 12. This is starting with Objective 6. This is going to be a little bit of a longer video, so as always, I encourage you to take breaks in the middle where you see fit. So we'll start with Objective 6, and Objective 6 says to describe how a nerve impulse is initiated and propagated along a neuron. And so neurocommunication and muscle excitation is based on the idea of electrophysiology, which is essentially cellular mechanisms that produce electrical currents and electrical potentials. So let's get definitions for electrical potential and current. An electrical potential, the definition here is the difference in the concentration of charged particles between one point and another. This is a form of potential energy, or energy that has the ability to do work. And electrical potentials can produce a current. An electrical current is a flow of charged particles from one point to another. Okay, let's move on. So living cells, your neurons are polarized. They have a charge difference across the plasma membrane. And that charge difference is called the resting membrane potential. From here on out, I might signify that as RMP. The RMP of your living cells is about -70 millivolts in a unstimulated, what we call resting neuron, hence the resting membrane potential. That negative value means that there are more negatively charged particles on the inside of the cell membrane than there are on the outside of the cell membrane. And so let's talk about how we get this resting membrane potential. There are no free electrons in the body, as you would find in an electrical circuit, say when you turn on a light in your house. So therefore, the electrical currents in the body are actually created by the flow of ions, especially sodium and potassium, through what are called gated channels. Gated channels, okay? So the resting membrane potential actually is a result from the combined effect of these three factors. The fact that the ions will diffuse down their concentration gradient through the membrane, the fact that the plasma membrane is selectively permeable, which means it allows some ions to pass easier than others, and the fact that cations and anions have an electrical attraction for each other. So first, we'll talk about a potassium ion. A potassium cation, remember, positively charged. They have the greatest influence on the resting membrane potential. So the plasma membrane is more permeable to potassium than any other ion. So I want you to imagine a scenario where a cell has all of the potassium in its intracellular fluid inside the cell. And along with the potassium in the ICF, the cell also has a lot of anions, or negatively charged ions, that are impermeable to the membrane. They cannot escape due to both their size and their charge. We can think of things like proteins, RNA, ATP, etc. So the potassium ions have the ability to diffuse through what are called leak channels, essentially just channels where they leak through slowly. And those are found in the plasma membrane. And ions, those potassium ions, diffuse down their gradients and into the extracellular fluid, leaving those anions behind. As a result of this, the intracellular fluid gets more and more negatively charged because the potassium, the positive charges, are leaving. And as the intracellular fluid gets more negative, it's actually going to start to pull back some of that potassium, right? Because of the opposite charges. So eventually what you'll get is an equilibrium, right? And an equilibrium in this sense means that there's no net movement of potassium anymore. It's not going out, it's not going in, it's going in and out at the same rate. But essentially, we can think of that as no movement at all. At that point, at equilibrium, potassium would be 40 times more concentrated on the inside of the cell than on the outside. And so if we stopped our scenario right here, where potassium was the only ion that had any effect on the resting membrane potential, the resting membrane potential would actually be about negative 90 millivolts. So obviously that's not the case in reality, right? Because we said negative 70 millivolts is the resting membrane potential. Because now we have to talk about sodium ions. Sodium is about 12 times stronger in the extracellular fluid as it is in the intracellular fluid. The resting plasma membrane is just much less permeable to sodium than it is to potassium, but the sodium is still able to diffuse down its concentration gradient into the cell because it is attracted by the anions in the cell, the negatively charged ions. The sodium leak is, only a trickle. It's just a little bit, and it's much less than the potassium leak, but it is enough to cancel out some of that negative charge inside the cell, and it reduces the voltage across the membrane. Okay, so that's why you get it down to about negative 70 instead of negative 90. Now, this leak is happening all the time. So to combat this, the cell uses a lot of energy to power these sodium-potassium pumps. And every time this pump takes 3 sodium ions out of the cell and pumps 2 potassium ions into the cell, this takes 1 ATP, 1 cell dollar. So by removing these more positive charges than it brings in, this actually contributes minus 3 millivolts to the overall resting membrane potential. So this is how we fight that leak that's happening. The actions of these pumps alone account for about 70% of the ATP used by the nervous system. So even though we say the cell is at resting membrane potential, the neurons are actually highly active in order to maintain the resting membrane potential. They're essentially waiting for something to happen, but they're not resting in the sense of they are not doing any work. Okay, now let's talk about changes in this membrane potential, and I want to give you some definitions. At rest, a neuron is considered polarized, and that is, of course, when the inside is more negative than the outside. When that membrane potential inside the cell increases or becomes more positive, this is called depolarization. Then, when the resting potential is returned to normal, we call this repolarization. And when the membrane potential inside the cell decreases and becomes more negative than normal, we call this hyperpolarization. So those are some definitions to keep with you as we move along in the rest of this chapter. Okay, let's talk about local potentials. So when a neuron is stimulated by something, let's say mechanical pressure, chemicals, heat, etc., a local disturbance in membrane potential is caused. It's just a disturbance that's happening in a very specific area. So typically this response begins at, you can see here in the diagram, the dendrites. And it spreads through the soma, down the axon, and ends at the axon terminal. So let's imagine a neuron is chemically stimulated, and the chemical stimulant, also called the ligand, will bind to a receptor on the neuron at the dendrites. This opens what are called ligand-gated channels, and especially ligand-gated sodium channels. These channels open and allow sodium cations to flow into the cell. Not leak anymore, but flow. This sodium inflow obviously cancels some of the internal negative charge of the intracellular fluid. And so the voltage across the membrane drifts towards zero. So once again, this is called depolarization, when you have a voltage shift to a less negative value. That incoming sodium will diffuse for a short distance along the inside of the cell, and the process here, what happens is it creates a wave of excitation. This short-range change in voltage is called a local potential. Now, there are 4 characteristics that distinguish local potentials from the action potentials we're going to talk about later. So these will make a little bit more sense when you learn about action potentials after this slide. The first is that local potentials are graded, meaning that they vary in magnitude depending on how strong the stimulus is. They are also decremental, which means that they get weaker the farther they spread from the point of stimulation. This is good so that it prevents long-distance effects of just a local signal. They are reversible. If a stimulation ceases cation diffusion out of the cell, it'll quickly return to the normal resting potential. They can be excitatory or inhibitory. So that means that some of the neurotransmitters will make the membrane potential more negative, hyperpolarization, and it'll become less likely to produce an action potential. That's inhibitory. Everything we've talked about up to this point has been excitatory. Okay? Depolarization occurs when the positive ions enter the cell. This is usually sodium. And hyperpolarization occurs when the positive ion leaves the cell, usually potassium, or sometimes negative ions entering the cell. Again, depolarization of the cell is excitatory. Hyperpolarization is inhibitory. All right. Now we'll talk about action potentials. Action potentials are a more dramatic change produced by the voltage-gated ion channels in the plasma membrane. When the local current arrives at the axon hillock, it depolarizes the membrane. Now, for anything more to happen, that local potential must rise to a very critical voltage called the threshold, and it's negative 55 millivolts in your neurons. This is the minimum voltage needed to open those voltage-gated ion channels. The neuron, once that happens, the neuron now fires or produces what is called an action potential. Because at that threshold, the voltage-gated sodium channels open quickly, and all of this sodium rushes in. Now, the gated potassium channels open much slower, and we'll talk about what happens to them later. So, thus, the initial effect on membrane potential is due to the influx of sodium. Initially, only a few sodium channels open, but as more and more sodium enters the cell, further depolarization occurs, and more channels open. This is, of course, a positive feedback loop. Okay, so what we've talked about so far is that a local potential increases the resting membrane potential to negative 55 millivolts, which is called the threshold. At that point, the neuron fires and the voltage-gated ion channels open and depolarization occurs. This occurs until about plus 30 millivolts. You can see up here, all the way up to about plus 35. At that point, the sodium channels close. So as this rising potential passes 0 millivolts, those sodium channels get inactivated and they begin to close. And by the time they all close and the sodium ions stop flowing in, the voltage peaks at approximately plus 35 millivolts. The membrane is now positive on the inside and negative on the outside. So it's important to know this means the polarity has been reversed. By the time the voltage peaks, those slow potassium channels that we talked about are now fully open. And what happens now is potassium ions are repelled by that positive intracellular fluid, and now they exit the cell. And their outflow repolarizes the membrane, which means you see this shift back to negative. This is a negative feedback loop. The potassium channels stay open longer than the sodium channels, so more potassium will leave the cell than the sodium that entered. Therefore, this membrane potential, the voltage here, actually drops to 1 or 2 millivolts more negative than that original negative 70. This is an overshoot, and it's called hyperpolarization. Now, we don't reach an equilibrium because we have those sodium-potassium pumps that are quickly going to reestablish the resting membrane potential. Okay, so here's what we've seen so far. There is a depolarization stimulus. That means that the voltage-gated sodium channels open, sodium diffuses into the cell, and more depolarization occurs. Now we'll talk later about a depolarization stimulus. Now let's talk about a refractory period. And there are two types of refractory periods. The absolute refractory period, which is the time in which a second action potential just absolutely cannot be initiated. This is due to the fact that those voltage-gated sodium channels are inactive during this time when repolarization is occurring. They have to be reset before the membrane can then respond to the next stimulus. That's the absolute refractory. Then there is a relative refractory period. The relative refractory period is the time period during which a second action potential can be initiated with a very strong stimulus, a supra-threshold stimulus, just a very strong one. In this case, the potassium channels are open and the sodium channels are closed, and the membrane is still hyperpolarized, but if you get a very, very strong stimulus, a secondary action potential can be initiated. This brings us to the all or none law. Once threshold has been reached, an action potential will happen. A stronger stimulus will not produce a stronger action potential because action potentials, unlike local potentials, are not graded. So what you can see here is that on this x-axis here, the stimulus gets stronger and stronger as you go to the right, but the action potential, all of them have the same amplitude. So this one did not meet threshold potential. This one finally does, and it still reaches the same even when the stimulus gets stronger. Action potentials are also non-decremental, meaning that they do not get weaker with distance. The last action potential at the end of the nerve is just as strong as the first action potential at the beginning. And also action potentials are irreversible, so if the neuron reaches threshold, that action potential goes to completion. This is the all or none law. How do we increase the response? So a stronger stimulus will not make a stronger action potential, but it will make action potentials occur more frequently. This is called frequency modulation. So you can see a stronger stimulus now makes many more action potentials. A stronger stimulus may also activate more than one or a few neurons. This is called recruitment of neurons. If something is extremely hot, more neurons will be fired so that you get away much quicker. Think of it that way. Okay, now let's talk about the conduction of nerve impulses. This is the ability for neurons to communicate with each other. And if a neuron is able to communicate with another cell, it means that the signal that it's sending has to travel to the end of the axon. And there are two separate ways in which this happens. The first is we'll talk about what happens in an unmyelinated axon, and then we'll talk about what happens in a myelinated axon. Okay. So in unmyelinated fibers, this is called continuous conduction. It's a relatively simple case. Note, this action potential does not travel along the axon. Rather, what's happening here is that one action potential stimulates the production of another action potential. Okay, so an unmyelinated fiber has voltage-gated channels along the entire thing. So when an action potential occurs, sodium enters the axon, diffuses a short distance. This excites the voltage-gated channels that are distal to the action potential. Sodium and potassium channels open and close, and by repetition, the next distal membrane gets excited as well. And this is a chain reaction that occurs until the signal reaches the end of the axon. Now, remember, what is happening here is that action potentials are occurring one right after the other, kind of like dominoes. So we can distinguish action potentials from nerve signals. The nerve signal is the traveling wave of excitation that's produced by the self-propagating action potentials. So again, I said it's kind of like falling dominoes. No one domino or action potential reaches the end of the line, but each one falls into the next, right? That's the nerve signal reaching the end, not a single action potential. Okay, now we can talk about conduction in a myelinated neuron. Again, myelin is something that provides insulation, and it improves the speed of the axon's cable properties. So myelinated fibers, they conduct signals in a very different manner than continuous conduction. So this is called saltatory conduction, down there in pink. These fibers that are myelinated cannot conduct a signal in a continuous mode because the voltage-gated ion channels are too scarce where their myelin is. And plus, the myelin insulates the fiber from the extracellular fluid anyway, so the sodium wouldn't flow into the cell. It couldn't. So therefore, where the nodes are of myelin, action potentials can't occur. And the nerve signal has to require some other way of transversing the distance from one node to the next. So the important difference here in saltatory conduction is that depolarization occurs only at those little nodes of Ranvier. There are a high density of those voltage-gated ion channels in the nodes. The current is carried by the ions and it flows through the extracellular fluid from node to node. And so what occurs is you'll see action potentials that appear to leap from node to node. What happens, what occurs here is that this greatly increases the speed at which signals are propagated down the line. And this also allows for the signals to be sent over greater distances without any degradation. So action potential conduction speed is increased by the diameter of the neuron, because this reduces resistance to the spread of charges, and also myelination because of saltatory conduction. Some examples: a thin unmyelinated neuron might send a signal at 1 meter per second, where a thick myelinated neuron could send it at 100 meters per second. So obviously a huge change. Okay, now we'll move on to Objective 8, and we'll talk about neurotransmitters. So the first thing I want to talk about is that in the early 20th century, biologists sort of assumed that synaptic communication, or the communication between two neurons, was only electrical. And that seemed right because neurons seem to be so close to each other that they must be touching. And so the signals were sort of transmitted electrically like normal wires. But through more careful histological examinations, it was revealed that there is about a 20 to 40 nanometer gap between neurons. So this gap is called the synaptic cleft, or the synapse, the space between adjacent neurons. And so when this first was discovered, some people said that cannot be true. However, in 1921, a German pharmacologist, Otto Louis, conclusively demonstrated that the neurons can communicate by releasing chemicals. And so what he did was he knew that the vagus nerves supply the heart and other organs with these neurochemicals to help slow it down. And so he opened up 2 different frogs and flooded their hearts with saline solution. He then stimulated the vagus nerve of one frog and showed that its heart rate dropped. He then took the saline solution only from that frog and squirted it into the heart of the second frog and showed that the solution alone reduced the frog's heart rate. That was evidence enough that that solution now contained some sort of chemical that stimulated the heart to slow down. Now, what he discovered was actually a neurotransmitter called acetylcholine. This is the first known neurotransmitter, and it's something we'll talk about in just a little bit. So, once the nerve signal reaches the end of the axon, it can go no further. So this must trigger the release of the neurotransmitter. It stimulates a new wave of electrical activity and across the synapse to the postsynaptic neuron. The first neuron in the signal path is the presynaptic neuron, which releases the neurotransmitter. The second is the postsynaptic neuron, the one that responds to the neurotransmitter. Let's talk a little bit more about synapse. Presynaptic neurons may synapse with a dendrite, a neurosoma, or axon of the postsynaptic neuron. And in that order, they form axodendritic, axosomatic, and axoaxonic synapses. A neuron could have enormous numbers of synapses, right? It's like a web. It has to talk to a lot of different outcomes. So a spinal motor neuron is covered by about 10,000 axon terminals from other neurons, 8,000 ending on the dendrites, and about 2,000 ending on the neurosoma. In the cerebellum of the brain, one neuron could have as many as 100,000 synapses. Here is an example of what we just talked about. On top here, there's the soma, the axon terminal, and the space between is the synapse. Then we have the axodendritic synapse, where the axon synapses with the dendrites of another axon, an axosomatic, where it's synapsing with the neurosoma, and an axoaxonic, where one axon synapses with another axon. Okay, the axon terminal of the presynaptic neurons contains synaptic vesicles containing neurotransmitter. Many of those vesicles are docked on release sites, ready to be released. And then there's a reserve pool of synaptic vesicles located further up the membrane, and it just sort of goes, once you release some, more are ready to be released down the line. A postsynaptic neuron will contain proteins on their membrane that function as receptors and ligand-regulated ion gates. Okay, let's talk about types of neurotransmitters. Acetylcholine is the first one. Well, first of all, there are 100 neurotransmitter types, and there's 4 major categories. The first is acetylcholine. Acetylcholine is formed from acetic acid, which is acetate and choline. The second are amino acid neurotransmitters like glycine, glutamate, and GABA. 3, the monoamines. They're synthesized by replacing the carboxyl group in the amino acids with another functional group. The catecholamines, which is epinephrine, norepinephrine, and dopamine, and the indolamines, which are serotonin and histamine. The fourth category are the neuropeptides. We'll talk more about those on the next slide. 5, purines, especially adenine, can also be a neurotransmitter. And 6, gases like nitric oxide and carbon monoxide. Let's talk a little bit more about neuropeptides. Neuropeptides are chains of 2 to 40 amino acids that are stored in the axon terminal as these larger secretory granules called dense core vesicles. They act at lower concentrations but have longer lasting effects. Some released from the non-neural tissue, like gut-brain peptides, will cause food cravings, and some actually function as hormones. Others modify the actions of neurotransmitters. Some examples here: endorphins and cholecytokinin. Okay, let's talk about synaptic transmission. Synapses vary. Some neurotransmitters are excitatory and others are inhibitory. And sometimes the transmitter's effect differs depending on the type of receptor on the postsynaptic cell. Some receptors are ligand-gated ion channels, and others act through secondary messenger systems. So next we'll talk about and consider 3 kinds of synapses: the excitatory cholinergic synapse, the inhibitory GABAergic synapse, and the excitatory adrenergic synapse. Okay, here is the cholinergic synapse that uses acetylcholine. Basically, what you should take away from this is that acetylcholine excites some postsynaptic cells and inhibits others. But we will talk about the excitatory function. And so what you see on the right here is that you'll see that the nerve signal arrives at the axon terminal and it opens voltage-gated calcium channels. The calcium enters the knob and triggers exocytosis, right? Calcium's entering the knob here, and it triggers exocytosis of acetylcholine. The acetylcholine diffuses across the cleft and binds to the postsynaptic receptors. You can see the green binding here to the receptors. The receptors are ion channels. They open and they allow sodium and potassium to diffuse. The entry of the sodium, of course, causes depolarization in postsynaptic potential. If this depolarization is strong enough, it's going to cause an action potential at the trigger zone. Okay, now let's talk about the inhibitory GABAergic synapse. Amino acid neurotransmitters such as GABA, they work through the same mechanisms as acetylcholine, like we just learned. They bind to ion channels and they cause immediate changes in membrane potential. So GABAergic synapse employs gamma aminobutyric acid, or GABA, as its neurotransmitter. This nerve signal triggers the release of GABA into the synaptic cleft. The GABA receptors in this example are chloride channels, and so chloride ion enters the cell and makes the inside more negative, not more positive, than the resting membrane potential. And we know when that occurs that the postsynaptic neuron is inhibited and is less likely to fire. An excitatory adrenergic synapse employs the neurotransmitter norepinephrine, also NE, and also called noradrenaline. NE and other monoamines and neuropeptides. They act through a secondary messenger system such as cyclic AMP or CAMP. The receptor in this example is not an ion channel, but is a transmembrane protein associated with another protein called a G protein. Let's take a look at what that looks like right now. So if you look at number 1, that's the unstimulated norepinephrine receptor bound to the G protein. And then in number 2, you see a binding of norepinephrine to the receptor, which causes that G protein to dissociate from it. That G protein then binds to adenylate cyclase, which is an enzyme that converts ATP to cyclic AMP, the secondary messenger. Cyclic AMP then can induce several different changes within the cell, and then 5 through 7 shows the possible different outcomes. Number 5 shows potential depolarization. Okay? All right. So in order to stop a neuron from signaling, you have to do 2 things. 1, you have to stop adding new neurotransmitter to the synapse, and you got to get rid of what is already out there. Because you want a reaction to occur, and then you don't want it to. So imagine if you touched a hot pot of coffee, you want to remove your hand, but then you don't want to keep moving your arm, right? You perform the action, you want to stop now. So getting rid of the transmitter that's out there is a very important step, and it can be achieved in 3 ways. Now, the first thing to do is stop releasing new neurotransmitter. That's easy enough to do. How do we get rid of the neurotransmitter that's out there? The first is through neurotransmitter degradation. And what happens there is that an enzyme comes around and breaks down the neurotransmitter into fragments that no longer have any stimulatory effect. Our example here is acetylcholine gets broken down by acetylcholinesterase, which is just an enzyme that breaks down acetylcholine. And that way, that acetylcholine can no longer be effective. The second is that it can be reuptaken in another way. So the axon terminal can actually reabsorb the neurotransmitter through endocytosis. Therefore, it's no longer in the synapse and no longer has a effect. And then the third and the final way is through just simple diffusion, where the neurotransmitter simply diffuses away from the synapse. And in this case, what you'll find is that the astrocytes in the central nervous system absorb it and then return it to the neurons for reuse. Okay? The next thing I want to talk about is neural integration. Neural integration is the ability to process, store, and recall information and use it to make decisions. Chemical synapses allow for decision making. The brain cells, right? The ability to process, store, and recall memories is through chemical synapses, and the brain cells are incredibly well connected, allowing for really complex integration. In fact, if you're reading this slide, you're using pyramidal cells of the cerebral cortex, which have about 40,000 contacts with other neurons. The trade-off through chemical transmission is that there's a little bit of delay. It's not as fast as it would be if you were just using an electrical symbol and there was no synapse. Now, neural integration is based on the types of postsynaptic potentials produced by the neurotransmitters. So once again, it's the first point here. It just says exactly what I said, that this integration is based on the postsynaptic potentials occurring in a cell receiving chemical signals. So for a cell to fire an action potential, we know it has to be excited to its threshold level, typically negative 55 millivolts. So an excitatory postsynaptic potential, or EPSP, is a voltage change from the resting membrane potential to threshold. So an EPSP usually results from the sodium flowing into the cell. Some chemical messages that we talked about, like GABA for instance, inhibits that postsynaptic cell by hyperpolarizing it. This is called an inhibitory postsynaptic potential, or an IPSP. And that occurs when the cell's voltage becomes more negative than it is at rest, so it will be less likely to fire. It can result from chlorine entry, chloride entry, or potassium exit from the cell. Okay, summation, facilitation, and inhibition. So one neuron might be receiving input from thousands of other neurons. Some incoming nerve fibers could produce EPSPs, and others could produce IPSPs. And so that neuron's response basically depends on adding up the EPSPs and IPSPs and seeing which wins, excitatory or inhibitory. So summation, like I said, is the process of adding up those potentials and responding to the net effect. And this occurs in the trigger zone. So here's an example of this. Suppose you're working in the kitchen and you accidentally burn yourself on a hot pot. The EPSPs in your motor neurons will cause you to jerk your hand back quickly so you can avoid being burned. Yet, a moment later, you go grab a hot cup of coffee. But since you're expecting it to be hot, you don't jerk your hand away from it. Because at some level of the nervous system, the IPSPs prevail and inhibit you from jerking your hand away really quickly. Because at some point in the nervous system, you have learned that this is not going to burn you. It's not going to hurt you, and so you can keep your hand on it. It's really quite interesting. So the balance between those EPSPs and IPSPs enables that nervous system to make decisions. Temporal summation occurs when there's a single synapse that generates those EPSPs so quickly that each gets generated before the previous one fades, and it allows those EPSPs to add up over time to a threshold voltage that triggers an action potential. Spatial summation occurs when the EPSPs are from several different synapses and they add up to threshold at one axon hillock. Several synapses will emit enough sodium so that threshold is reached. The presynaptic neurons collaborate to induce the prosynaptic neuron to fire. And this is an example of facilitation, which is a process in which one neuron enhances the effect of another. So, to do this mathematically, a typical EPSP is a voltage change of only about 0.5 millivolts and lasts only 20 milliseconds. So if a neuron has a resting membrane potential of negative 70 and a threshold of negative 55, you need at least 30 of those EPSPs to do so. And there are two ways in which they can do that so that the action potentials occurs. The temporal summation or the spatial summation. Here's what that looks like. Here on number one, on top is intense stimulation by one presynaptic neuron. So a lot of stimulation from one, or you could have simultaneous stimulation from several of the presynaptic neurons. Either way, you just need to add up enough stimuli so that the action potential can occur. Okay, one more thing to talk about, a few more slides, is presynaptic facilitation. This occurs when one presynaptic neuron enhances another one. It's the opposite of what we'll talk next about inhibition. So neurons routinely work in groups to modify each other's actions. So obviously presynaptic facilitation is where you have one neuron that helps to facilitate another neuron. So up in top there we see neuron F and neuron S. The S is the signaling neuron and F is the facilitating neuron. So the neuron F releases serotonin, which makes the voltage-gated channels in the S axon terminal stay open longer, so that the EPSP happens more often. Presynaptic inhibition is the opposite, where now instead of F, we have neuron I, that when that fires, it suppresses the S neuron, and it reduces or halts the unwanted synaptic transmission. It inhibits the neuron, cell I, inhibits them by releasing GABA. And this prevents those voltage-gated calcium channels in the axon terminal from opening, and so the knob will release little or no neurotransmitter at all. It's inhibitory. Okay, and 2 slides left. This one I'm talking about neurons that work together. They often work together in groups of thousands called neuron pools. And just like if you were thinking about a circuitry of wires, neurons work in circuits as well. And there's 2 types, a diverging circuit and a converging circuit. In a diverging circuit, an individual neuron sends signal to multiple downstream neurons. So an example of this is one motor neuron in the brain can stimulate thousands of muscle fibers in order to move your leg or your arm or close your hand. In a converging circuit, the input from many nerves is funneled into fewer and fewer. An example of this is your brainstem has a respiratory center. The respiratory center receives signals from thousands of other converging signals, and it can process them and make decisions about respiratory rate and the respiratory pattern. And the last thing here to talk about is anesthetics and proprioception. So when we talk about anesthetics, general anesthetics, which put you out and when you need a surgery or something, many of those general anesthetics work by blocking neurotransmitters. So just something interesting to think about. And then proprioception, which is just knowledge about where you are in space, is dependent on information that's mediated both by neurotransmitters via receptors and the sensory nerves that are embedded in the tendons. So with that being said, guys, I will see you in the next video.